Welcome to another webinar at DeLong Discussions. My name is Ahmad, and I'm an applications engineer at DeLong America. The subject of today's talk is imaging nanoparticles using a benchtop low-voltage electron microscope. Okay. This is an overview of today's talk. I'm going to start with some basic introduction about electron microscopy, followed by more in-depth detail about low voltage electron microscopy, advantages of a benchtop TEM applications, and followed by finally a live demo as promised. So electron microscopy, I'm sure that these concepts are familiar for you or for most of you guys, but as a basic summary, the electron beam has a shorter wavelength than a visible light. Therefore, we are able to resolve infinitesimal objects with higher resolution at higher magnifications. The electron beam interacts with our sample and produces a number of different emissions, including backscattered electrons, or BSE in short, X-rays for EDS, forward scattered electrons, FSE, and electron diffraction. So in TEM, we are mainly interested in the beam, the portion of the beam that transmits through our sample. Therefore, we need a thin specimen that's electron transparent somehow, so our beam can penetrate through, and we can get the beam on the other side of the thin specimen using the camera or the fluorescence screen in some older devices. TEM is great for high resolution imaging. It's good for morphology. It's great, and it's great for internal structures and to perform size measurements. Another famous mode of electron microscopy is scanning electron microscopy, or SEM in short. SEM uses a focused beam in the specimen as opposed to the parallel beam, as opposed to the wider beam that we use in TEM. So we focus our beam into a tiny region, tiny spot on our specimen, and we raster along some lines to form the image. So we can also call this focused beam the electron probe. In SEM, we are not collecting the beam on the other side of the specimen, so we don't necessarily need to have a thin specimen. We can rather image bulk samples, and so really we don't care about the thickness as long as our instrument, as long as our chamber dimensions allow for it. SEM is great for surface morphology. It's good for topography, and it gives you the ability to see the depth of field. And it's great for compositional contrast if you are using the backscattered electrons, the BSE, because there are different modes of SEM. And so again, with SEM, we are able to see different heights in our sample. As you can see in this image, we are able to get different sort of information. So we can achieve information and high details about the surface. We can see in here, we can see different pores, we can see different cracks, structures, and everything on the surface of our sample. Also, the intensity, the light intensity, or the contrast enables us to get information about our sample's composition. So in BSE, in backscattered electron microscopy specifically, the lighter or the brighter regions are more electron dense, more dense, so they can emit more backscattered electrons. Therefore, we can get some information about the density and the compositional contrast, basically the composition of our sample. Scanning transmission electron microscopy, or STEM, is another mode. It's basically a mixture. It's a combination of SEM and TEM. It's like SEM in that it uses an electron probe or a focused beam, and it's like transmission electron microscopy, or TEM, in that we need a thin sample so that our beam penetrates through, transmits through, and we collect it using the STEM detector on the other side of the specimen. So we need thin sections, thin samples. It's great for high resolution imaging. It can provide high contrast, especially for light elements. When we talk about TEM, that's the sort of image that comes into mind. Massive instruments, underground labs, anti-vibration platforms, chillers or liquid nitrogens for cooling down the column. And as you can see in this image, the red arrows, so the one on the left is the liquid nitrogen, the one on the bottom right is for the anti-vibration platform. Therefore, all of this adds to the cost of maintaining and providing the infrastructure for such big high voltage TMs. Not to mention the scheduling difficulties, and we have to share it with other researchers at the core facility. Another challenge with TM is the beam damage. Beam damage can be categorized into three main types, three main categories: knock-on, radiolysis, and heating effects. Later, I'm going to talk about carbon and show that the knock-on damage can be reduced, especially by lowering down the voltage. And on this animation, we can see part of our sample before illumination, 10 seconds after illumination, and 2 minutes after illumination. We can see some changes, some different chemical reactions happening in here, basically damaging our sample. Because high voltage can penetrate through our samples, because in TEM, the high voltage cannot produce satisfactory contrast, especially for light elements. Therefore, in some biological applications, we are forced to increase the density of our sample through adding heavier compounds such as uranyl acetate. These staining procedures are toxic, and at the end of the day, we are altering the chemistry of our sample. So it's another challenge in terms of sample preparation for high voltage. So let me ask you guys, what electron microscopy mode or modes do you use in your research? So please select all that apply in the next 30 seconds. So TEM, SEM, STEM, and the last one is electron diffraction. So whenever we have TEM, we can also obtain electron diffraction patterns to infer information, to understand information about the crystallinity of the sample. I can see that answers are coming up. Maybe it's enough to stop the poll right now. Most of the attendees, they use TEM and SEM. 70%, 65% SEM, TEM, less STEM, and smaller portion electron diffraction. Okay, perfect. So that's, let me close the poll in here. With LVEM, with low voltage electron microscopes, we can actually achieve all of these modes in the single device. With that, let me go to the next chapter of this next chapter or next topic of this presentation: low voltage electron microscopy. So why low voltage electron microscopy? Why LVEM? The main two reasons for acquiring and using a lower voltage beam is basically that it enables you to increase image contrast, especially for low atomic number materials. And because by reducing the accelerating voltage, we can reduce column size and produce such miniaturized benchtop TMs. So a big advantage is the increased contrast. And then the second main advantage is being able to produce such small instruments and increase the accessibility. On the diagram on the left, we see cross sections for electron scattering on the vertical axis, and then on the horizontal axis, we see atomic number compared at different voltages. As we decrease the accelerating voltage, the scattering cross section increases in our material. Therefore, the contrast increases, and by reducing the voltage, as I said, we can reduce the column size, and therefore increase the accessibility of TEM. Here I did a simulation, Monte Carlo simulation, for a 100 nanometer thick carbon. The top one is at 5 kV and the bottom one is at 100 kV. So different accelerating voltages, low voltage and a high voltage instrument. At the top, on the top one, we can see that we have much more scatterings. And whereas in the lower one, we see that as if our beam, is just penetrating through, it's just transmitting through our sample without any scattering. In order to produce a good contrast, we need higher scatterings. On the right diagram, again, it's a similar concept, so cross-section versus electron energy. The yellow and black dotted lines, the yellow and black dotted curves represent inelastic and elastic cross-sections, respectively. And the red dotted line is our voltage that I'm talking about today, 5 kilovolts. So again, at lower voltages, the cross-sections, the coefficients for inelastic and elastic scattering are higher. Once again, the higher the cross-section for these scatterings, the higher the contrast. By lowering the voltage, we can increase the contrast. So on the left images, I have 3 different micrographs of DNA origami. So DNA origami is made up of light elements, basically carbon, hydrogen, oxygen, phosphate. So the one on the left is at 5 kV, the middle one is at 25 kV, and the right one is at 100 kV. For these images, I draw a line, 500 nanometer, and calculated the light intensity between white and black ranges, and made a graph out of it. As you can see, the gray value range is higher for the lower voltages, whereas we can clearly see in the gray box that the contrast is lower. The contrast range is lower for the higher voltage. Another challenge is beam damage. So on the right, you can see protein damage with beam energy. As we increase the beam energy towards the top, we can see that we are breaking some chemical bonds of our protein sample. Another big advantage, as I said, is increasing the accessibility. So having a benchtop TEM. So by the way, it's worth to mention that this is the world's only benchtop TEM. So having a benchtop, using a benchtop TEM, you can have it in your lab, can be installed in your own lab across that bench or the corner of your lab. So you can have routine access to it. No need to book, no longer need to book the core electron microscopy facility. And you can basically image your samples whenever you want. Simplified sample imaging flow is, of course, preparing your sample. Then you need to book the core EM facility. Then you have to bring your samples to that facility whenever the time permits and whenever you have that appointment. And finally, image your samples, only to find out if your sample was prepared well enough in the beginning to begin with or not. But with an in-lab electron microscopy instrument, you can prepare your sample just as in the same way as you always prepare your samples. And then once it's dry and ready to be inserted into the ultra high vacuum column of the TEM, you can image it basically right away. So if it's a good sample, then you continue imaging. If it wasn't a bad sample, then you didn't waste so much time. You haven't wasted so much time. You go prepare your sample in a better way, and then you can come back to image them. So basically, it increases the throughput of your work. Here at DeLong Instruments, we have 2 main line of products. One is LVEM 25E, the other one is LVEM 5. LVEM 25E operates at 25 kilovolts, and it's our more advanced microscope. But the subject of today's talk and demo is LVEM 5. So in LVEM 5, we use an accelerating voltage of 5 kilovolts for TEM, STEM, and SEM, as well as electron diffraction. And with that, let's dive deeper into the advantages of having a benchtop LVEM 5 or a benchtop TEM. So this instrument was introduced some 20 years ago. It's an entry-level system. The resolution for TEM is 1.2 nanometer. For SEM, 4 nanometer, and for STEM is about 2.5 nanometer. So nanomaterial analysis, biological analysis, all of those samples can be imaged with LVEM5. And again, it's a great addition to your lab, inside your lab, in lab electron microscopy. So the sample preparation is exactly the same. So whether you need to sonicate the solution or basically all those pre-deposition steps are the same. You deposit your sample in the same way. You pipette the number of microliters that you always do. And then once it's dry, you can insert it into the ultra-high vacuum column of this benchtop TM. One added benefit is removing the necessity of staining. So in lower voltage electron microscopy, in low voltage electron microscopy, staining is only optional. And once again, the resolution is great. It's nanometer resolution. So it can go as high as 1.2 nanometer. It's a benchtop instrument. So these are some dimensions. So you basically need a nice and sturdy table that can, so that you can put your microscope on the top. The dimensions of the table are 1.2 meter by maybe like 62 centimeter. And the total height is around 1.5 meters or something like that. All you need, the only infrastructure that you need, it's basically not an infrastructure, it's only a wall outlet. That's it. So no need for anti-vibration platforms or coolers, chillers, liquid nitrogen whatsoever. Only a single 24-hour 24/7 electricity. So it's small, it's compact. Everything you see in here is run through this, is using the same outlet. So on the top we have the microscope along with the console and the monitor, and at the bottom we have the turbomolecular pump, the electronics unit, and the PC. So these are some specifications again, which I've already covered most of them. So it runs at 5 kV. The resolution is at 1.2 nanometer. The magnification can go as high as 700,000 times with magnification boost. And these are some other numbers. With that, let's move ahead and talk about some applications. So one of the great applications is morphology. What characterization techniques do you use on a regular basis? So it's another poll. Please answer the poll in the next 30 seconds or so. So once again, what characterization techniques do you use on a regular basis? TEM, DLS, AFM, or other techniques? DLS is dynamic light scattering. AFM is atomic force microscopy. I'm happy to see that most of the attendees here are using electron microscopy the most. So let's share the results. So yeah, so almost 3/4 are using electron microscopy. Some others DLS, AFM, and others. Perfect. Let's stop the poll. Okay. So as a summary, AFM, DLS, and TM. So AFM stands for atomic force microscopy. AFM is great for profiling the surface, the topography. It's a great measurement of height. Therefore, you can measure the surface roughness, but it's limited in the X and Y dimensions. So again, it's very exact. It's very good in terms of the height, but in terms of X and Y, in terms of the 2D shape of your sample, it lacks resolution. DLS, on the other hand, can provide a good scattering measurement. We can analyze particles in solution. It's non-destructive and it gives a good average particle size in a short amount of time. But again, it lacks the shape information. and it assumes spherical particles. So it basically assumes that all of these particles are spherical. And also it's sensitive to aggregates. So we are not really sure if this is an aggregate of our particles or just a bigger particle. TEM, on the other hand, enables you to see your sample directly, so to visualize your particles. So you can directly measure the size, you can see the shapes, and also you can see the internal structure within your sample. The only limitations, if you can say, is the height measurement. So it's not so good in the Z space, but it's very good in terms of X and Y 2D dimensions. So these images and the ones that I'm going to show next have all been acquired using the LVM5. So on the left, we have gold nanoparticles, and on the right, we have ytterbium. So we can see the contrast is good. We can define define individual particles. And I spent maybe like less than 2 minutes on ImageJ, free software, open source, to do a basic thresholding and do some basic size measurements. It's good, and I can still spend probably more time to manually threshold each sample and select those particles. But what if we have more complex samples? So if I want to do the same thing with ImageJ, it's very laborsome, and I have to spend a lot of time manually selecting my particle. But that's when image processing comes into rescue. And for this, we actually used Image Pro. It's a third-party software, and with that, we could easily define our particles, let the software detect them, measure them, and provide all of those different measurements. So area, diameter, total object counts, etc. Having this luxury of having different modes in the same instrument enables us to do correlative microscopy. So, for example, on this image, I have iron oxide samples. So in TEM, I can understand about its 2D information, so shape, distribution, measurements, etc. And I can see the same iron oxide sample in SEM to understand about its surface, the shape in 3D space, and as well as its compositional contrast. So again, we can compare these images. We can acquire both of these images to have a broader, more in-depth understanding about our sample, all by using a single instrument. On the image on the left, I have some lead sulfide. We can see the outlines of the particles. We can see some of them are round in 2D or probably spherical in 3D. And some of them are darker, some of them are less dark. They are more gray. If they are darker, that means they are more dense or thicker, so that less electrons are transmitting through. Whereas if it's white or bright, such as in the case for the carbon background, that means it's more electron transparent, or more electrons can just penetrate through, can just pass through. On the image on the right, I have a combo sample. I have gold nanoparticles, I have latex, and a carbon nanotube. This is a sort of sample that I usually use in my trainings because it's fun to work with, and my trainees or the soon-to-be electron microscopists like to perform TEM in the low mags first to be able to see the large latex particles, and then as they progress, they can achieve nicer images at higher magnifications to be able to see the gold nanoparticles at a better magnification. I just mentioned the carbon nanotube. Speaking of carbon is very important in electron microscopy. It has a dominant presence in organic matter in all biological compounds, and it's also present in a variety of inorganic materials. All of this only says how important it is to work with carbon, basically. So the sensitivity of carbon into contrast is of utmost importance for us. And it's something that lower voltage can excel at. So in these images, I have some carbon nanotube. On the left, I have the single multi-wall carbon nanotube. We are able to see the walls, the intersection. And once again, I have a carbon nanotube on a carbon film. So that's why I said the sensitivity of carbon to the contrast and everything is very important. And that's something we excel at. On the right, I have the SEM image of the same carbon nanotube. Thanks to the contrast of this backscattered electron microscopy technique, we are able to see the brighter spots, which are gold nanoparticles embedded in these nanotubes. We are good in imaging carbon single layers, such as graphene. So again, on the left, I have a TEM image and a TEM micrograph. The background is a carbon film, and the slightly darker gray area is a single layer graphene. Because of the contrast, we are actually able to see the sheet of the carbon that is folded on top of each other, or we can see all those wrinkles. On the right, I have an electron diffraction pattern. So again, when we have TEM, we can easily get those electron diffraction micrographs as well, and use this to understand more information, gain better information about the crystallinity of our sample. Soft matter is another area that low voltage excels at. So in here, I have some quantum dots. We are able to see their shape and morphology and distribution. And the last but not least, biological samples. So biological samples are made up of low atomic weight materials. Low voltage provides a better contrast inherently. So in here I have adenoviruses. On the right image, it's a TM image. I have these adenovirus particles on top of a carbon film. And on the left one, I have these adenoviruses in a cell. So the left image is a thin section, basically. Same thing, bacteriophage and tobaccoviruses, good contrast, good resolution, easy to measure different aspects of our sample. And in fact, we are able to see the tinier details on the tails of these phages. DNA origami is another emerging field in biotechnology. On the left, I have some 2D sheets of DNA origami, and on the right, I have some three-legged DNA origami structures. We are able to see the distance between each layer, and we can measure those layers one by one. This is Professor Armin Delange, the namesake of our company. He's the founder of electron microscopy in the Czech Republic. His mission was to make electron microscopy accessible for everyone. And that's a quote from him. Every scientist should have the opportunity to regularly use a transmission electron microscope for his or her research. And that's our goal too. These are some proud LVEM users across the world, working in different fields and disciplines. And just a brief summary of what we said in today's webinar. So main reasons for low voltage electron microscopy: the ability to increase image contrast, especially for low atomic number materials. And also it allows for the use of permanent magnet lenses instead of electromagnetic lenses, which allows us to reduce the column size. It can be a direct replacement for traditional high voltage electron microscopes. So only a fraction of the cost of the big ones. Low maintenance, minimal maintenance and service is needed. Ease of installation, so it's very compact. It can be installed in short time, the training is very easy, and everything can be wrapped up in just a couple of days. And it doesn't require any infrastructure, only needs a wall outlet. That's it, with 24/7 power. LVEM 25 and LVEM 5 both can have TEM, SEM, STEM, electron diffraction.